Good morning to Inchi Aslan. My name is Thanuja Kumaran from DBT3B and my metric number is 23DBT20F1047. So today I would like to do the virtual lab on gram staining. Gram staining is used to detect the presence of the bacteria and sometimes fungi in a sample taken from the site of a substance infected. So now I would like to start the virtual lab on gram staining. So first the procedure: heat the inert solution loop for sterilize purpose and then take the sample and gently place on the slide. Again heat the inert loop for sterilize purpose and then let the slide dry 5 minutes. Float the slide with crystal white for 1 minute. After 1 minute, rinse with water. And then again float the slide with iodine for 1 minute. And rinse the slide using water. After that, decolorize with alcohol for 5 to 10 seconds. And then rinse again with the water. And then float the slide with saffron for 1 minute. And again rinse using the water. After that, heat the slide gently under the Bunsen burner. Keep moving the slide so that the sample will not discharge, and then view under the slide. Next is catalyzed test. Catalyzed test is a particularly important test used to determine whether a gram-positive Cauchy is Staphylococcus or Staphylococcus. Procedure: first, drop water on the slide and then take the sterilized stick and pick an isolated clone and then dissolve it in the water on the slide. And then watch the result. After that, throw the stick into the biohazard waste box and then again drop water on new slide and using sterilized stick pick an isolated clone and then mix it in the water on the slide. Again, throw the stick into the biohazard box and then dispose the slides into the disposal box. Is Colgillus test. Colgillus test identifies whether an organism produces the exoenzymes Colgillus which cause the fibrin of blood plasma to clot. So now we do the experiment. The procedure is: first drop the water on the slide. After that, using the sterilized stick, again pick an isolated clone and then mix it in the water on the slide. After that, throw the stick into the biohazard box and then using the dropper, drop the rabbit plasma on the slide. After that, gently rock and mix the suspensions. Watch for clumping or collection to develop slowly. After 15 minutes, again drop the water on the new slide and then using the sterilized stick, pick an isolated clone and mix it with the water on the slide. After mix it, throw the stick onto the biohazard waste and again using the dropper, drop the rabbit plasma on the water and gently rock and mix the suspensions and watch for the clumping or collection to develop slowly. At last, dispose the slides into the disposal box. Now, oxidize test. Oxidize test is used to identify bacteria that produce cytochrome C oxide, an enzyme of the bacterial electron transport chain. Pick the sterilized stick and pick an isolated clone and place on the dry slide. After that, observe the color change and again throw the stick into the biohazard box and repeat this step for 2 more samples. After that, dispose the dry slide into the biohazard box. That's all from me. Thank you.